Hello and welcome to this lesson on advanced higher biology, focusing on SQA revision support based on the guidance that the SQA is given. This is a Forth Valley and West Lothian Regional Improvement Collaborative lesson and it is of March 2022. By the end of this lesson, you should know how to answer the 5-mark extended response question. You should be able to know how to answer either of the 9-mark extended response questions, and you should know how to answer question types that have been suggested by the SQA. By the end of this lesson, you should be able to construct an essay based on the information that the SQA have provided to you. So the SQA's updated guidance as of March 2022. So what will be in the exam? Well, for some courses, the SQA have published important information on which broad topics, contexts, or content will or will not be in this year's exam. In biology, we've not had anything taken out, but we have been told what the key areas are for the essay questions for the 5-mark and for the 9-mark the extended response questions. For some courses, they have been given study guides to help them answering questions, and these give tips and hints, and these are on the SQA's website for you to look at. And in a small number of courses, we've not been given them in biology. However, there are short study notes which can be used. The best place to use or find study notes for advanced higher would be to look on Scholar. Scholar has really good notes for advanced higher biology. So, throughout this lesson, you'll see various symbols on the slides to indicate what you should do, and here are the symbols which may be used and what they mean. So if there is a listen icon, that literally just means to listen to what is being said in this particular slide. Speak and repeat is repetition work. Read is to read the content on the slide carefully. Write/copy is to take a note of what is on the slide for your personal notes. The tick is to correct your work after a task. The pause icon is to pause the video to complete the task, and there will usually be an indication of the time allowance for this. And the watch icon is to watch whatever is going on in that particular scene. The Advanced Higher Biology exam this year will be on the 19th of May. The duration of the exam is 3 hours in total. The exam is worth a total of 100 marks, of which 20 marks are in the multiple choice section A and 80 marks are in the short answer section B. There will be 2 extended response questions in the exam, one of which is worth 5 marks and the other worth 9 marks. The 9-mark question will give you an option of 2 choices to choose from in the exam. The 5-mark question will not offer a choice. So we're focusing on exam content that we've been told by the SQA. So we're going to focus on the 5-mark essay question. And the 5-mark essay question is on the following key areas. It's on Unit 1, Key Area 4, Control and Signalling. In particular, we've been told to focus on hydrophobic signalling and the control of transcription. So required content for this 5-mark question. The kind of information that you need to know is the hydrophilic signaling molecules, how they move and how they reach their target and bring about a change. So hydrophobic signaling molecules can diffuse directly through the phospholipid bilayers of membranes and so bind to intracellular receptors. So they travel through the cell membrane or the phospholipid bilayer to be able to reach the inside of the cell and inside the cytoplasm. Many hydrophobic signaling molecules bind to a receptor when they're inside the cytoplasm or the cytosol, and the receptor complex then moves to the nucleus. So when they've bound inside the cytoplasm, they'll then start to move towards the nucleus of that cell. The receptor proteins for steroid hormones are transcription factors, and they're going to bring about a change to the rate of transcription. As they move from the cytoplasm to the nucleus, they bind to these areas known as their specific DNA sequences within the cell, and they're known as hormone response elements. So when they bind, when that hormone receptor complex with our hydrophobic signal and its receptor bind to these hormone response elements, this causes influence to the rate of transcription. So it can either bring about an increase in the rate of transcription, or it can halt the rate of transcription. It depends on what type of steroid hormone has been released at that particular length or that particular part in time. Examples of steroid hormones include things like testosterone, estrogen, cortisol, and thyroxine would be another example of a steroid hormone as well. I would advise taking a note of this slide for your own personal notes. So, an example question that you might get asked is, give an account of the action of hydrophobic signaling and give an example of a signaling molecule. Now, this would be a 5-mark question. So you may want to pause the video. We're going to work on the 5-mark extended response question, and you'll be given 8 minutes to continue working on this task. So take the 8 minutes, pause the video, and then come back to the next portion of this video. So you should have taken the 8 minutes that you were told to take. So we're looking at our given account of the hydrophobic signaling and given an example of a signaling molecule worth 5 marks. So I'd like you to mark your answer that you've written just now. Now, mine is just a rough kind of template of what an answer could be for this question. So just mark yours as appropriate just now. So hydrophobic signaling molecules diffuse across the phospholipid bilayer through the hydrophobic tail portion. And that would be where the first mark would be. So saying something about how the signaling molecules diffuse across the cell membrane or the phospholipid bilayer or the plasma membrane. These signals then enter the cytosol and can bind to intracellular receptors. This would be where our second mark could come from. You could say how they travel into the cytoplasm and bind, or you could say how they diffuse and bind to their receptors in the cytoplasm. That would be your second mark. You could also mention that this occurs in the nucleus as well. When the signal binds to its receptor, it moves into the nucleus, and specific regions of DNA known as hormone receptor elements, HREs, are where it binds. This would be where the third mark would come into play. This then allows the increase in rate of transcription, or it can halt the rate of transcription. This would be the fourth mark. Now you could say something about how the alteration of the rate of transcription, anything along those lines would get you this mark in this instance. An example of a hydrophobic signal is something like testosterone or estrogen or cortisol or thyroxine. These are all examples of steroid hormones that would get you the mark in this example question. So this question was worth 5 marks. And you can see clearly the 5 marks where you can get them in this example. So focusing on the 2 essay questions now. So we've got our 9-mark essay questions, and these are on Unit 2, focusing on sex and behaviour, and in particular it's focusing on reproductive behaviours and mating systems. In the next few slides, we'll go over information that you'll need to answer the essay question, the 9-mark essay question focused on mating systems and reproductive behaviours. So females have a lot to lose if they mate with the wrong male, and they're therefore selective about who they mate with. They look for certain criteria, and these include physical features, so size and strength, which confer dominance and so preferential access to resources. So a female or a male is looking for a female is looking for a male that is big and shows that he can provide food or other resources to her. She'll also look for behavioural features. So animals that are female will look for these behavioural features which may indicate willingness to invest or good parenting skills. Females will compete with other females for the right to choose the most desirable male, also known as the alpha male. They gain little from multiple matings, and females seek quality, not quantity, when they're looking for a mate. Almost every reproductively capable female will be able to find a mate of some sort. I would advise taking a note of the next few slides as well. Males are far less choosy as they have little to lose and everything to gain if they can have as many mating opportunities as possible. Males are not tied to rearing offspring and seek quantity. While they would prefer a superior female, if presented with a sexual opportunity, they will take it. Males compete vigorously with other males for access to females. Male reproductive success is however varied, and a small number of males will achieve many mating opportunities, usually known as our alpha, while many males will never mate. Mating systems are based on how many mates an individual has during one breeding season. These systems range from polygamy, polygyny, and polyandry to monogamy. And these are the 4 terms that you need to know for the essay question as well. Polygamy is when individuals of one sex have more than one mate. So a particular sex has more than one mate. Polygyny is when one male mates exclusively with a group of females, and this can be seen in populations like lions would be a good example of that. Polyandry is one female mates with a number of males in the same breeding season, trying to increase her chances of reproductive success. And monogamy is probably the one that you're most aware of from everyday life, is the mating of a pair of animals to the exclusion of all others. And this kind of breeding system can be seen commonly in humans in some instances, and it can also be seen in ducks. Ducks are an example of an organism that are monogamous. So an example question for you again for the 9-mark question, using the information that we've just looked at in the previous slides. Give an account of reproductive behaviours and mating systems in animals. Pause the video, 9 marks. So we're working on the 9-mark extended response question A from the previous slide, and you'll be given 15 minutes to work on this slide. So to go over our example question, so give an example of reproductive behaviours worth 4 marks and mating systems in animals worth 5 marks. So females have a lot to lose during mating. They look for males that are physically impressive and show particular behavioural traits. Males have less to lose from mating and seek quantity. That would be our 2nd mark. So our 1st mark would come from saying about how females are looking for physically impressive males and ones that show behavioural traits. That's the 1st mark. 2nd mark comes from the males have less to lose and seek quantity of mating opportunities. Sexual dimorphism is a method of sexual selection displaying fitness. This is where the 3rd mark would come in for the reproductive behaviours. These features do not provide a physiological advantage, but a breeding success advantage, and it is a display of health to the female. Mating systems are based on how many mates an individual has in an individual breeding season. This is where the 5th mark would come in, and it's the 2nd part of our essay question we're looking at now. Polygyny is an example of this, where one male mates exclusively with several females. Monogamy is when a pair of animals mate exclusively of all others. Polygamy is when individuals of one sex have more than one mate. And polyandry is the final mating system with one female mating with several males in a season. So 9 marks in total there. I would use this as a kind of basis to mark your own essay question against as well. So the second 9-mark essay is in the following topic, still Unit 2, but we're focusing on evolution, and in particular, the SQA have asked you to focus on drift and selection for this particular question. So, to continue with our essay question, focusing on evolution this time and our drift and selection, we'll go through the required information for this area and make sure that we have the content that we need to be able to answer the question. So, during evolution, changes in allele frequency can take place through the processes of either genetic drift, which is a random process, or through selection, which is a non-random process. And selection can come in two forms, either natural selection or sexual selection. Natural selection is the non-random increase in frequency of alleles which are favourable to the organism, and the non-random decrease in the frequency of the deleterious alleles. So basically, those alleles that allow a species to survive will be passed on, and those that are not an advantage and those that are disadvantage to the particular species will not be passed on. Natural selection is achieved by survival of the fittest. You've probably heard this way back in National 5 and in higher if you did higher biology. Fitness refers to those organisms which have alleles which will increase survival. Organisms which are able to adapt are also genetically fit. Sexual selection is the non-random process involving the selection of alleles that increase the individual's chances of mating and producing offspring. Sexual selection does not increase chances of survival, but just the likelihood that the organism will reproduce and then pass on its alleles or pass on its particular genes that offer an advantage. This particular method may lead to sexual dimorphism. So sexual dimorphism being the differences between males and females in a particular species. It might be appearance, or it might be size, some sort of factor, colour, these kind of things. Examples of sexual selection are things like male-to-male rivalry, which can be seen in things like giraffes or walrus, and female choice, which would be something like licking, which is part of this particular unit as well. So, other information we need to know. So we need to know about our genetic drift. So genetic drift occurs when random events cause changes in allele frequency from one generation to the next generation. Genetic drift is more important in small populations, as alleles are more likely to be lost from that gene pool. So you need to know about the importance of bottleneck and founder effects when you're talking about genetic drift. So population bottlenecks occur when a population size is reduced for at least one generation. So it could be that a random event such as an earthquake separates a species. What happens is that population size would be decreased because of that earthquake, and it creates a kind of bottleneck for the passing of genes. It kind of slows the movement of these genes. Founder effect is also another means of genetic drift, and basically founder effect occurs through the isolation of a few members of a population from a larger population. So if we have a group of birds, say, and we have another earthquake and it causes the splitting of an island, or it causes these particular birds to fly off to a different area, but only a small proportion of them go there. What we might start to see is over time there might be differences in the number of alleles. It might be different combinations than what the original larger population that they came from have. So the gene pool of the new population, the smaller population, is not an accurate representation of the original gene pool. So this new group, who might be, show mutations or different combinations of alleles, are not a representation of the original gene pool. A gene pool is altered by genetic drift because certain alleles may be underrepresented or overrepresented, and allele frequencies change in this. So what we see is that genetic drift brings about this random change, either overrepresenting particular genes, a gene that might provide an advantage, or if they're separated, it might be that that particular gene is a disadvantage in that area, and it might be underrepresented because it doesn't confer some sort of advantage to that species. So I have another example question for you, another 9-mark question. So whilst you're doing your practice, come back to this slide and pause it on this slide. So we're going to look at describing the process of drift and selection for 9 marks. So we're working on the 9-mark extended response question B, and you'll be given 15 minutes to continue working on this question. Okay, so back from our 15 minutes of working on our question. So our question again was describe the process of drift and selection, so our 9-mark question. Now try to mark your answer against this. Obviously yours will not be word for word the same as mine, so just try your best to be able to mark it against this answer here, this model answer. So selection can either be not random or non-random. Natural selection is an example of a non-random selection. That's where our first mark would come in. So we've named that process. This is the increase in frequency of alleles that are favourable to an organism. This is done by survival of the fittest, so our third mark coming from there. Sexual selection is another form of non-random selection. That's our fourth mark. It involves the passing on of alleles that will convey an advantage to an organism for successful mating chances. Genetic drift is a random process which can result in an increase or decrease in frequency of alleles and is best seen in small populations. That's our 6th mark. Chance events such as earthquakes or other natural disasters bring about genetic drift. 7th mark there. Population bottlenecks occur when a population size is reduced for at least one generation. Finder effects occur through the isolation of a few members of a population from a larger gene or a larger population, and that's our 8th mark. So mentioning our population bottleneck and our founder effect are two methods of genetic drift. The gene pool of the new population is not representative of that in the original gene pool. A gene pool is altered by genetic drift because certain alleles may be underrepresented or overrepresented and allele frequencies change over time. So that would be where our 9 marks for this question come into play, and I would like you to try and mark yours against what I have here. So different question types that the SQA are trying to make you more aware of. So the first one that they've said is focusing on described questions. So these questions require you to state what's happening in a situation or an experiment. So an example question from a past paper I have here is describe how protein electrophoresis is used to separate proteins. So focusing on that particular word at the start of our question. So describe. So we need to state what is happening in this particular method of separating proteins. So pause the video just for 30 seconds or for however long it takes you to answer this question, and we'll go over the answer in the next slide. So the kind of thing that you would want to put as your answer, they've said charge or current flowing through a buffer separates proteins based on the things like size, mass, shape, or charge. So what we can see here clearly from their answer is that they're focusing just on stating what is happening in this particular method. So they're just stating what happens in protein electrophoresis. So they're saying it separates proteins based on their size, their mass, their shape, or their charge. They're just stating what this is. Another type of question that you may be asked to think about in the exam is explain questions. Now these are questions where you must give a reason for what is happening in the situation that's posed in the question. So you kind of have to give some sort of justification as to why your answer, or it might be relating to a statement that they've given you, is the case. So we have an example question here where they have asked you to explain the term cooperativity in relation to oxygen binding to hemoglobin. So they've given us a scenario. They've said, relate this term, being the term cooperativity, to oxygen binding in hemoglobin. So we need to relate this situation to another situation is what they're wanting to do. So same thing again, pause this video here and have a go at this question, try to make an answer, just 1 mark answer. So we have got our explain question here and we've got our answer on the screen now. So we had to relate our answer to the situation of cooperativity and how it relates to the binding of oxygen in this situation with hemoglobin. So what we can see is binding makes the binding of other oxygen more likely. So the binding of one oxygen to our hemoglobin opens up the other oxygen binding sites of our hemoglobin. So what we can see here is that they're relating the information that they've given us to the answer that we are giving in that instance. We're taking the information they've given us and using that to in our statement kind of thing. Another area that the SQA has asked you to make yourself more comfortable with looking at is things like box plots. So kind of focus on data handling and box plots. Box plots may be something you might be comfortable with if you've done them in maths, or it might have been a while since you've visited using box plots. So you should be able to use and interpret these. So I've got an example question here. Now you can see, we'll just go over our box plot as is. So we can see in the diagram here, we've got our box plot. We can see it starts with our minimum value. So that's the very lowest value in a data set that we've been given. From there, we've got the median, which is the middle number, and we've got our maximum value. The maximum value is just the biggest number in the data set. So we've got the smallest number, the middle number, and the largest number. Now the lower quartile is that the average basically between, or the, sorry, not the average, the median between the actual median itself and the minimum value. So the one that is between the minimum and the median. So it's this point here for our lower quartile, or Q1 you might have been calling it in maths. Same thing again for our upper quartile. It's the same concept. So it's between our median and our maximum value. So it's essentially the median of these 2 points here, and it's our upper quartile. And it may have been known as Q3 to you in maths as well. So we've got 5 data points on our box plot, 5 values that we can interpret from our box plot. So this question here is focusing on a situation of heart rate variability. So let's have a go at this question. So short-term variations in the time interval between heartbeats is termed heart rate variability, or HRV. HRV is one measure that can be used to assess health. The box plots give information from one study about the influence of age and gender on HRV. From the data shown, which of the statements about HRV is correct? So pause the video, have a go, read through each of the statements that they've given you and try to come up with the correct answer based on what you can see in that particular box plot there. So the correct answer for this one here is that there is a trend towards lower HRV, or heart rate variability, in females. So we can see throughout the graph that there's a lower variability in females of the actual results, and we can see that from the results that are given here. We can see that the greatest error in measurements is for ages 25 to 35. Well, no, that's not true, because the measurements don't vary greatly. The mean HRV for females aged 35 to 44 is lower than 40 units. We can see that that one is not true as well. It's greater than that. And gender-related differences in median values are greater than age-related differences. So for D, we can see definitely that the gender differences are closer than the age-related. We can see that the age-related, there are bigger differences between them. Another area that we should be familiar with is in our calculations, and we should be familiar with this Hardy-Weinberg equation in preparation of your exam. So you should know this, you should be able to use it, and you should be using practice questions to get comfortable with using this. The HW principle can be used to determine whether a change in allele frequency is occurring in a population over time. And changes in this suggest that evolution is occurring. I would advise you to take a note of this. So our equation is p squared plus 2pq plus q squared equals 1. So all of these adding up together are equal to 1. That tells us, that tells us there's a steady rate of evolution going on. So the p squared stands for frequency of homozygous dominant individuals. So 2 big letters. Our q squared is the frequency of homozygous recessive individuals, so having 2 small letters, the weaker gene. And our 2pq is the frequency of heterozygous individuals, so those that have a mix of a dominant allele and a recessive allele. Here's an example question of a Hardy-Weinberg question. So the frequency of a given allele in a population is a measure of how common that allele is as a proportion of the total number of copies of all alleles at a specific locus. For a locus with one dominant allele and one recessive allele, the frequency of the dominant allele and the frequency of the recessive allele can be used to calculate the genetic variation of a population using the equations below. So P plus Q equals 1, so the frequency of allele A being the dominant allele and the frequency of the smaller allele. And we've got our Hardy-Weinberg equation here. If the allele frequency of the recessive allele is 0.7, the proportion of individuals that would be heterozygous is what? So either A, B, C, or D. Pause the video and have a go at this particular question just now. So the correct answer for this question is 0.42. So using your Hardy-Weinberg, you should have been able to work this one out. Another area that you need to be aware of for your advanced higher is experimental design questions. So looking at a particular experiment, how it's been conducted, and to be able to criticize if there's been problems with how it's been designed, and also to be able to state different things like confounding variables or like conditions or control groups or these kind of things. You need to be aware of how to answer experimental design questions. So thinking critically about how an experiment's been designed. You should also know the following terms to help answer questions about experimental design. And I would advise that you take a note of these. So reliability is when consistent values are achieved in repeats of an experiment. So when you do an experiment, you conduct it, it's the ability to achieve the same results time and time again. So when you're doing your experiment, that is when you would, if you repeat it or if you have multiple samples, that they are achieved each time. Validity is when our variables have been controlled so that any measured effect is due to the independent variable. So the thing that we're changing. So we're definitely confirming that if we are changing X, that it is X that is actually causing that effect, so that nothing else is attributed to our potential, effect in our study. Accuracy is data is close to the true values, so our true value might be our mean, and this would allow us to see that, okay, our values are coming in within this particular range, and that allows us to see that we are close to that particular true value. So each time that we do it, our values are accurate. Precision is when measured values are close together. So that a precise study would be something where the values would be maybe within 0.1 of each other. they're not varying too greatly from that true value. So something could be accurate but not precise. So an accurate study might have data that's close to the true values. It might be that the true value is 0.5, so you have some data that's 0.25, 0.6. However, that would not be precise. A precise study would be looking for it to be as close as possible to that true value. So it might be that for true value 0.5, our precise values would be like 0.47 or 0.49 or 0.48, like as close as possible to that true value in that situation. Another area that you need to be comfortable with is interpreting graph data. In advanced higher biology, you should be comfortable with interpreting data from graphs that contain at least two y-axes. And in these two y-axes, may be that they're either looking at the same thing, they might be using a different scale. You need to be able to identify and look at what units they're using, if it's per percentage or if it's in measurements in milliliters or millimeters. You need to pay attention to that, and you also need to pay attention to the scale. A good tip for interpreting data from graphs like this, if you get asked to state a value at a particular time or something like that, is to use two different highlighters. So to use one highlighter and to color the line that corresponds to a particular axis in that one color, and to highlight that axis that color so that you don't get mixed up, and then use your pen for the other one. So it might be that you have a yellow pen for the first one and an orange pen for the second one, and highlight the other ones, just so that you don't get confused at which line is which. It's very common that the lines look similar as well. It might be that you've got a dotted line and a solid line, but at particular points, just because of the way that the graphs are laid out, you might not be able to tell clearly which line is which. So it just allows you to differentiate easier between the two lines in that situation. You should ensure that you correctly read what each scale is, as they will not have necessarily the same units, and that's kind of something that we've touched on there. So, you've got an example question here of an interpreting graph data question, and we can see that we've got two very distinct lines here. However, not all questions will be so distinct. They will not always be so different, and they might have lines going across the graph as well. So firstly, let's just have a look at this graph. So we can see along the bottom, they're giving us years, and we can see it's going up in every 4 years. So it's every 4 years for each point in the graph and the years at the bottom. On the left-hand side, as we go up, we can see that this is cases times 1,000. So for every one of these, it's times by 1,000. So if it's 100, then it'd be times 1,000. It'd be 100,000 cases at that particular point. And we can see that our measles notification times 1,000, or our cases, is this kind of thinner line, the one that's the kind of jaggy line as well. On the other side, we can see the vaccine uptake percentage, and we can see that this is the solid line in this situation here, and it's in percentage. So on one side of our graph, we have got times 1,000 cases, and then on the other side of our graph, we can see that we've got percentage of people that are taking the vaccine for measles in this situation. So what I'd like you to do is I'd like you to have a go at this question here. So looking at this MMR. So MMR vaccine was introduced in 1988 to combat the infectious viral diseases of measles, mumps, and rubella. In 1998, a report published in a peer-reviewed medical journal claimed to establish a link between the MMR vaccine and the disorder known as autism. Although criticised heavily and eventually discredited, this research led to a considerable reduction in the number of children being vaccinated. In some areas, such as Swansea in South Wales, vaccination rates fell as low as 67.5%. Over the same period, a huge increase in the number of measles notifications occurred in that area. Explain how the events in Swansea confirm that the graph not only shows correlation, but also shows causation. So the question asked us to look at our the graph and to show that the data shows not only correlation, so that these two things, there was a link between them, but also that there was a causation. So as the answer scheme says, it says, linking it to the scenario that we've been given about Swansea, as vaccinations go down, the number of cases increases. So we can see that there's a correlation, but this is actually also the cause of this as well. So as we can see, the number of vaccines goes down, we can see that the number of cases goes up in that situation. Next steps for you and your advanced higher biology. So it would definitely be worth attempting the extended response questions that were in this presentation again, and each time try using your notes less and less, and to try and kind of gauge, how to answer the question. So it might be worth, occasionally looking at your notes, having them in front of you and putting a piece of paper over them to cover them, and only pulling that piece of paper when you feel that you need it. It gets to a certain point where you start to actually remember. Once you remember one point, it's kind of like the floodgates open, and you're able to remember the other points because it becomes methodical. So remembering the essay questions in that way will certainly help. It may be worth asking your teacher to create a mock style question of the whatever question it is you're working on for the extended response as well. I'm sure that in the coming weeks there will be other example questions that come about as well that teachers will be sharing with pupils. So it's definitely worth looking over the information for those extended response questions and attempting those to get used to them as best as possible. It's also worth ensuring that you revise problem solving type questions. So thinking about your interpreting graph data, thinking about using your Hardy-Weinberg equation, all of these different things, it is worth having a go at using past papers, practicing these techniques, getting used and getting comfy with difficult questions, standard questions that are problem solving questions. It would also be worthwhile looking at the usual questions that come up for these kind of problem solving questions. So it might be hemocytometer questions, using cell counting, getting used to, factorizing, working up your, making sure that you put your answers in scientific notation, things like that. It would also be worth looking at your questions that focus on dilution factors as well. My advice to you is just to use these past papers and to just get as much exam practice as possible. And best of luck from me.